So welcome everyone to the MICD, to this joint MICD MIDAS seminar. I am pleased to introduce Professor Ivo Dinov. He's a professor of the Department of Health and Behavior and Biological Sciences from the School of Nursing, and he's also a professor from Computational Medicine and Bioinformatics. He's the Associate Director for Education and Training for the Michigan Institute for Data Science, MIDAS, and the Vice Chair of the Department of Health, Behavior, and Biological Sciences. Professor Dinov is an expert in mathematical modeling, in statistics, and high throughput computational process and scientific visualization of big data. And some of his research applications are on neuroscience and biomedical imaging, among many other things that he applies his research. And today he will talk about analytics, data science, time complexity, and Spacekime that I'm really eager to learn about. So welcome and thank you again for being here.
Thanks very much, Mariana. It's a great pleasure, and thanks for joining. This is a wonderful seminar that I've attended very often, and it's really great to see everybody on. So today I'll present some of the work that some of my students and a colleague of mine, Milan Velev, at Burgas Technical University are working. This is based on an upcoming book pretty much with the same title. So by the way, before I start, for those of you that are interested in, the slides are actually available online. If you just Google soccer news and go to today's event, you see a PDF slide deck that might be helpful, I guess, if you're interested in following up some of these things. So in this talk would be divided in three fairly complementary parts, building, I hope, to something that might be exciting for some of you, which is at the bottom, the applications. But before I get there, I'll have to introduce, why are we doing this? A little bit about the motivation and some of the challenges that we see with biomedical, large and complex biomedical data. And then I'll spend the bulk of it would be, to some extent, technical. We'll present what complex time is all about. We'll discuss the space-time calculus, the mathematical foundations, then mention maybe one or two open problems. And then we'll go into the applications where I can show you hands-on how it's actually used for research. So let's start at the beginning. A justification of what are the analytical challenges when you handle big data sets. So we are all familiar with IBM's formulation of the 4 V's, the volume, variety, velocity, and veracity of these complex data sets. In our work, we've looked at many dozens of case studies, and we've actually tried to identify the proper characteristics or the dimensions of big biomedical data. And these are listed as the 7 rows here, the 7 different dimensions of big data. And corresponding to each of these, starting from size, complexity, and so forth, we present various specific challenges that are encountered literally on a daily basis by anybody who's dealing with these kinds of data. So, the large size presents a lot of difficulties with harvesting, manipulating, and management of these very complicated data sets. The complexity of the data requires development of various data wranglers that are able to deal with data that's not necessarily homogeneous, that's not homologous, and you still need to be able to make some sense out of it. The incongruency of the data dictates the need for developing harmonization and data aggregation tools. Many a times the data is actually multi-source. It arrives from devices and experiments and all kinds of other things that are not designed to interoperate. So representing jointly this multivariate data and modeling it is a very difficult thing. Oftentimes the data is multi-scale, so how do we interpret data that's on a macroscopic level? Think about meters, centimeters, millimeters, to the meso, micro, and nano scale of the same objects being monitored. This presents a bit of a hierarchical problem. Very often the data is actually longitudinal, it changes, it morphs with time, which requires dealing with some of these temporal associations that carry forward. And last but certainly not least, that these types of data are never complete. So we have to be able to do what's called statistical obfuscation. We need to do statistical obfuscation to protect sensitive information and so forth. And here's just one example. We've analyzed hundreds of Parkinson's disease individuals, and for each individual, we have tens of thousands of signature biomarkers, which are derived by looking at the imaging, looking at the genetics, looking at clinical observations, and various other phenotypic and demographic data elements. So with that justification of the need, so let's just spend a little bit of time on what is complex time and why is it useful for our purposes. So before I dive into this, I want to remind everybody the notion of the Fourier transform, which maps or translates spatial temporal signals into spatial and angular frequencies effectively. So I've stated the Fourier transform here for the most general space-time based data, where the arguments of the function are obviously space-time events that are anchored in terms of x, y, z, and t. And then the corresponding Fourier transform and its inverse are stated over here. One of the most important things to realize here is that the complex transform both acts on complex valued functions and returns are complex valued functions. And these complex valued functions have very nice representations in terms of the magnitude of the complex number, which represents its size, and its phase. So let me just give you one very kind of simple example here. And maybe if my app works now, I can actually show you this interactively. So we've developed an app that allows you to draw one-dimensional signal. So you go ahead and draw this signal. And then you can see the decomposition. Here is the Fourier decomposition in terms of the magnitudes on the row in the middle, and then the phases are on the bottom. So when I bring the mouse over, it kind of, and obviously I can manipulate each of these things and see the effect of changing that specific magnitude accordingly, and then seeing the effect that it introduces, or manipulating the corresponding phases. As you can see, as the phases go to the right, they become they correspond to different frequencies and so forth. And you can manipulate these things as well, and see the effects interactively, at least for one-dimensional signals. For higher dimensional signals, it is a little bit more involved. But if you have a Java-enabled browser, you can actually try this on your own. So diving a little bit, making the problem a little bit more interesting, I'm going to show you now a two-dimensional representation of the Fourier transform. So I'm going to start with two images. Well, one is going to be a schematic image of the Earth, and the other one is going to be a schematic image of Saturn. And what I'm going to do is I'm going to pipe both of them through the Fourier transform. I'm going to reconstruct or actually analyze through the Fourier transformations. We analyze the signal, we derive the corresponding magnitudes of the Earth image and the phases of the Earth image. And correspondingly for Saturn, we have the magnitudes in the Fourier domain and the phases. So the experiment that I want to run now is I want to see, well, what is the effect of these almost white noise fingerprint-like images that represent the phases of the corresponding time domain data, which is the original two images. So here on the bottom rows, I'm going to be reconstructing or synthesizing an image, a space-time image, from using the Earth magnitude, but instead of using the corresponding Earth phase, I'm going to use the Saturn phase, okay? And when you reconstruct this by the inverse Fourier transform, you get something that looks neither like the Earth nor Saturn, but it looks more Saturn-like. Now, if you also completely nullify the phases and just use the magnitudes of the original Earth Fourier transform, you can still reconstruct something that doesn't look like an Earth, but it looks like perhaps a planet, a distant planetary object. The same thing for the Saturn, right? If we use the Saturn magnitudes, but instead of using the correct Saturn phases, we're going to use the Earth phases. When we reconstruct it, the image kind of looks more earthlier. And if we nullify the Saturn phases and again reconstruct, we obtain an image that is far from either, and it has obviously some problems. So the important take-home message from this example is that these phases, they matter a whole lot in representing complex information, in this specific case, two-dimensional images. So we can't really ignore the phases when we have complex valued objects. So a little bit about the history. Almost 100 years ago exactly, the German mathematician Theodor Kaluza developed this really interesting math extension, which was very, very abstract at the time, of classical general relativity to 5 dimensions. And then he partnered with the Swedish physicist Oskar Klein. And at that time, in the early 1920s, Oskar Klein was right here at the University of Michigan. He was in the physics department as a visiting scholar for 2 or 3 years before he went back to Copenhagen in the 1925 or 26. But they published this paper that they essentially, again, extended the classical Minkowski's 4-dimensional space-time manifold to a new 5-dimensional manifold, where they argue that there is an extra dimension that is so small that it belongs to Planck constant, so it's not traversable. So in other words, if you have 2 spatial locations or temporal locations that are that far apart, you know, this extra dimension is folded so deeply in that it is not observable effectively. So what we are going to be doing is we're going to be extending that theory a little bit. We're going to be capitalizing on this notion to define complex time. And it's going to be, you know, purely mathematically. Complex time is defined as an Euler representation of any complex number. So R is going to be the magnitude, and phi, which we're going to call the phase, is going to represent, as it says up here, the angular displacement or an event direction. Now, the beauty of this time extension or complex time extension of the standard classical positive real time is that the positive real time comes to play exactly as the magnitude R of the corresponding time. So here is a schematic where the 3-dimensional spatial dimensions are compressed into a single horizontal line, and then you have the orthogonal 2-dimensional complex time, where, again, you can see events that occur at the same time, just like anywhere on this green circle, events would occur at the same time, but have different directions. And this is going to play a very important role for us. Now, when you go from the green circle to the red circle, obviously you have an increment of the longitudinal order of the event, or R increases. If you go from the red to the blue circle, you have a spatial displacement of the event, right? So we can annotate these pictorially in this simple extension. So what's the rationale, you may ask, of playing around with complexifying or lifting the dimension of time from the positive reals into the complex field? Well, the time is just a special case, as you can see from Kime, and it just corresponds to a phase, to a trivial null phase. And again, later on, you're going to see how this plays a significant role. Algebraically, the multiplicative group of the positive real time is a well-defined object, right? With respect to the multiplicative operation, it's got a well-defined identity and so forth. You can do the associative law of multiplication works and so forth. But time is not a complete algebraic field. So in other words, when you take the 2 classical operations of multiplication and addition, we have a little bit of a problem, because the additive inverse element actually can fall outside of that multiplicative group R+, which is our time space. So mathematically, it's a little unsettling to have an incomplete object. Second of all, is that equations like this do not have solutions in R+, which is time, or even R, right? So these equations obviously have imaginary solutions, and they require complex number representations. So that's kind of the mathematical justification. From physics, there's a lot of well-defined rationales for doing this. And if anybody's interested in it, I'm not going to go deep into this, but look up this book on the problems of time, just to see the various conflicts that arise with the positive real definition of time. From an artificial intelligence and data science perspective, I will show you that random sampling is going to play a key role here. We are going to argue that random sampling in space-time is effectively corresponding to a very clever sampling of the phase space for the same time magnitudes. Also, if there's time, I'll show you the Bayesian representation of Spacekime analytics. And then at the end, I'll show you some of the examples that pertain to tensor linear modeling of these Kime surfaces, which we're going to be deriving, that are the analogs of the space-time time series. So data that's longitudinally varying with time, we're going to be translating it to a more complicated or more complex, more enriched mathematical object called a two-dimensional manifold. And these manifolds are going to represent what's called Kime surfaces. So they're going to be, quote unquote, indexed not by time, but they're going to be indexed by complex time. So this leads to, as you can imagine, some kind of fairly novel analytics. So it's not surprising, I suppose, that there is multiple parameterizations of the complex plane that can be used for different kind parameterizations. And, so just to make it a little bit more formal, I guess, the Spacekime manifold is defined, the dual nature of the Spacekime manifold as a holistic object is defining what K events, and these K events are complex events that are specified by where do they occur in space, by their point localization in space, and by their moment, but the moment is defined again, remember, if you think in a complex plane diagram that we had earlier, in that complex time notion. So having that, you can define the generalization of space-time intervals that generalize the Minkowski 4x4 metric tensor into a 5-dimensional tensor. In its most general form, it looks like this. Obviously, we're going to be interested in the flat Euclidean Spacekime manifold, where the signature of the metric is plus, 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 3 pluses for the space, and 2 minuses for the corresponding time-like or Kime dimensions. And this leads to intervals that are space-like, so that they're spatially separated, events that are light-like, that corresponds to trivial Spacekime intervals, or events that are not time-like, but Kime-like, that correspond to negative intervals. Whoops, sorry. Okay, so let's now look into the Spacekime calculus. I beg your pardon, please. Okay, there we go. So we can define Spacekime calculus by using what's called Wirtinger derivatives of higher dimensional arguments. And so here is an example of formulating the first order Wirtinger derivative with respect to different bases, right? The conjugate basis, the polar Kime coordinates, you can define it in Cartesian, obviously, and so forth. And obviously, here is the second order Wirtinger acceleration, if you wish, or the second derivative with respect to Kime. Now, once you have obviously the notion of differentiation in that space, you're interested in, well, can I do the inverse? Can I integrate? And indeed, integration can be defined pretty much the same way by extending the path integral for these complex functions. You can define the path integral in the complex plane, and this naturally leads to what's called the definite area integral over a specified domain, the indefinite integral, as well as operators like the Laplacian, for instance, here stated in terms of the conjugate pair coordinates. So we have this notion again of the ability of doing Spacekime calculus. Other extensions that we can introduce is we can take Newton's equations of motion in the space-time domain and transform them using the corresponding Wirtinger velocity and acceleration to, as it says here on the top right corner, here are the Newton's equation in motion in this in terms of time, in terms of the two-dimensional complex time. You can come up with a whole bunch of other generalizations of space-time. One of which is the Lorentz transformation between two reference frames, K1 and K prime. So the Lorentz transformation is important because it leaves the interval that I showed you a little bit earlier invariant with respect to this transformation. So we can define this as a second order tensor in terms of the proper coefficients. We can also do a number of other generalizations that relate to physics. For example, the law of additions of velocities can be generalized, the energy momentum, and so forth. One example that I'll just show you is the little image that's animating on the right-hand side. This is just an example of the ultra-hyperbolic wave equation with Cauchy initial data conditions in 5 dimensions, except that now the three-dimensional space, physical space, is compressed into a two-dimensional plane. So each of these planes, the xy on this three-dimensional framework here, the xy represents the quote-unquote three-dimensional space, and then the vertical axis represents one of the time dimensions, namely it represents the phase. It goes from minus pi to pi, maybe difficult to see here, but it goes from minus pi to pi, and the actual animatory, the motion that you saw, the animated motion, was actually the second time dimension, which is actually corresponding to the magnitude or to the longitudinal displacement of the event. So I know this is kind of difficult to picture, but nonetheless we can solve equations and actually visualize them using, albeit simplified representations. Now a little bit of a clarification here, and this has to do with ultra-hyperbolic wave equations are well known to not permit solutions, or if there exist solutions, the solutions are not guaranteed to be unique, stable, or global, right? So this is well known for ultra-hyperbolic equations. Now, the way that we are able to solve this is by introducing what's called non-local constraints. And with these non-local constraints, so here is the corresponding wave equation that sets equal on the left-hand side the spatial Laplacian. On the right-hand side, you have the temporal Laplacian, and the initial Cauchy data conditions are stated right up here on the right-hand side. So it turns out that you can obtain, in fact, stable local solution over a frequency region. So in other words, we're going to introduce these non-local constraints in terms of in the frequency domain, okay? And subject to these constraints, these solutions that are induced to these second order partial differential equations are actually stable and well posed. Now, let me just show you early on, I mentioned that one of the AI and data science related justifications for complexifying the notion of time is to introduce an alternative to the classical random IID, independent and identically distributed sampling. So this is again another app that we've developed a number of years ago. If you have a Java-enabled browser, you can see it. So what's happening here is we've drawn with a mouse a little distribution, and there is a curtain that doesn't really allow you to see the distribution. But what you're trying to do is, the reason why we do samples, the reason why we do measurements, the reason why we do observations is to make predictions or explicate the properties of the process that's behind that distribution, which we don't necessarily see explicitly in its holistic state. So what we do is we take these samples. So here is random samples drawn from this distribution, right? We draw random samples once, we draw random samples two. That's what's happening right now with COVID, right? We take a lot of data about how people respond to either vaccines or various other treatments that we have, and then we do summary statistics. So you see the last two rows, I know this is kind of difficult to read, but I'll just explain. So this is the unknown, sometimes, you know, unexpected distribution. Here is the actual samples, and now the last two rows represent for me the sampling distribution of one characteristic, in this case, the mean, the sampling distribution of the mean. And here on the bottom is the sampling distribution of the dispersion of my samples, right? So these are important quantities. Because these are the first two moments, right? Some people may be interested in the second, in the third, fourth, and higher moments, and that's perfectly fine as well. So we want to do something very similar here, but we are going to do this in the time phases. So here is a simulation where we are showing, see, I'm randomly walking through three different processes, the black, the green, and the red process. Each of these processes have a well-defined, for me, predefined mean. The green distribution is slightly positively biased. The green, beg your pardon, the black distribution is slightly positively biased. The green distribution is unbiased, it's got a mean of 0, and the red distribution is negatively biased. And we're taking a whole bunch of samples through that sample space. So the radii that radiated these lines that were coming off of the center of this circle, the magnitudes of them correspond to the longitudinal order. At what point in time was this measurement corresponding? And the phase distribution, the circular phase distribution, corresponds to the multiple instantiations of the observations. So what we're trying to do here is, if you get anything from this, I guess, presentation, I'm hoping it's the fact that you can do data analytics by observing either observations the way they natively occur on the left-hand side, and that's what we've been doing for thousands of years. Or you can do randomly sampling the phase space and then aggregating these phases with one or two observations alone to actually reconstruct a huge sample that can be AI or machine learning processed in some kind of a way. Now, in that framework, you can imagine there is a lot of open problems. I'll just mention one of these. It pertains to what's called ergodic theory. So let's just look at one example of how this thing works in practice. So suppose you're looking at an integrable function, and for our practical purposes, we're going to assume that this is a velocity function. And these integrable functions f, they're obviously defined at every location, x and t, right? You give me an event location, at what spatial place and at what time am I measuring a function, an integrable function like the velocity. Now, the Minkowski space-time x is a measure space, and I assume I have some kind of a measure defined on that space. And then I also assume that I have an operator, t, that maps the measure space x onto itself in such a way that it acts as a measure preserving transformation. So I have a transformation that doesn't stretch, shear, alter the significantly the input. So I have a measure preserving transformation, and a pointwise ergodic theorem in this specific case would basically argue that if I have my velocity and I compute the expectation at a given moment in time, so I fix the moment in time, and I compute the expectation by averaging across all the entire spatial domain, R3, I average, which basically means integrate against the measure of the space, your velocity function. So I'm going to call this f bar for the spatial averaged, and this is obviously a function of t. And if I do the opposite thing, if I fix a point in space, just one spatial location, and then I go ahead and integrate, quote unquote, the transformation applied. So where is this spatial location going in time 1, time 2, time 3, time 4, etc., all the way up to infinity? I'm going to call this f tilde. So ergodic theorem says that if these two things are equal, then you have ergodicity properties of the system. So in other words, spatial averaging versus temporal averaging leads to identical things, right? So this is not currently known about space-time. So we still need to derive the fact that space averaging, it would be equivalent to Keim averaging to argue that ergodicity might or might not be in play when you talk about space-time. So now, halfway through, you're probably wondering, well, where is the data science? Where is the neuroscience? Where is the biomedical applications? And you'd be right to do so. Now, by the way, in the chat, if anybody is already pretty bored, in the chat, I put a link to the dynamic. We actually have a pretty nice dynamic website where you can go through and explore some of these things with interactive visualizations and so forth. But coming back to our justification now, so we're going to go from the mathematical physics that we presented into the data, AI, and neurosciences. So let me draw some parallels. So particles that we talked about, we're talking about velocities of particles, we're talking about energies of particles, whatever. So these particles, they become objects, okay? Observable variables or dynamic variables in physics, they're going to become features in our data science framework. States of particles become datum, unique quantitative or qualitative values or instantiations of these features. Systems, particular systems, they become problems or data systems. And the most important one is that the wave function that we typically define in physics, this becomes in our data science domain, the inference function. Transforms, they're preserved. Transforms are transforms, whether it's Lorentz transforms or log transform, it doesn't really matter. States of systems become data sets, computable systems become computable objects, and so forth. So just to give you one really specific example to contextualize that mapping, the wave function is typically a function that solves the following second-order partial differential equation. So any function phi in terms of x and t that solves that equation. I showed you a little bit the most general way, and specific some of these wave packets that are in this specific form are known to be solutions. So you have wave functions that solve these equations. Now I'm going to be translating that notion of a problem, solving the equation here, to a problem fitting, for example, a generalized linear model or fitting an SVM classification model. These are the problems. And I want to know, well, what is the corresponding, not wave function, but inference function? So for the simple linear case, the inference function is very simple, right? An example of an inference function, because there may be many multiple inference functions. One of these is you can use ordinary least squares to compute the effect size estimates. And then your inference, of course, your inference function is essentially the one that leads these parameter estimates into testable statistics, and obviously ultimately to the corresponding probability values. In the non-parametric non-linear case, when you talk about support vector machine classification, pretty much the same thing happens. You have an inference function which is very directly related to the kernel, this obviously leads to again another inference that can be applied. In other words, where should that support vector machine division that separates or clusters the points, how should it be drawn, and how does it actually lead to the ultimate labeling that I'm interested in? I want to know, is this point labeled A and this point B, or are they part of the same cluster? So these are just two examples to motivate this. So we are going to be doing space analytics, Spacekime analytics. So you see, we're not the first ones to look into this. Crystallographers for over 50 years have dealt with this problem, the problem of the missing phases. So in crystallography, you basically have a small crystal that represents some kind of a protein or some kind of a substrate that you want to know the structure of. You bombard this with very high energy particles, and on the back, you see a projection. Now that projection simply reflects the magnitudes of the corresponding crystal. It represents the magnitudes, but the phases are unknown. So what people do is they either use prior information about the phases, or they use some kind of an experimental reproducibility strategy to actually reconstruct the three-dimensional structure of the quote-unquote protein using two-dimensional projections of the corresponding magnitudes. So this is not a new idea. So we're going to be doing exactly the same thing. We are going to be having, this dichotomy between space, space-time and Spacekime, and we're going to be going back and forth using various transformations, whether the Fourier Transform or the Laplace Transform or other transformations, to be able to either estimate the missing phases or derive some computed time estimates that can be used ultimately to lead to some analytics. So in this specific example, I'm just showing you a representation of a two-dimensional fMRI slice across time. So we're just looking at the isosurfaces. So we're looking at a three-dimensional perspective view of the isosurfaces of not really a four-dimensional fMRI, but just a two-dimensional cross-section of the fMRI across time. And we're not looking at the whole signal because the whole signal is going to be very dense. I need to use volume rendering. So we're just rendering the isosurfaces. Here is the reconstruction that only uses time by negating, if you remember in the very first beginning where we did the Earth and the Saturn images, how I negated the phases. And the image reconstruction did not look very pretty, very nice. So that's exactly what's happening here. We have a no phase reconstruction on the left, and then a perfect reconstruction with the correct phases on the right. And you can see that even though these isosurfaces look fairly homologous, there is a lot more fine detail that you can see in the correctly reconstructed image as opposed to the phase deficient reconstruction on the left. So now let's kind of transition a little bit into the analytics. So what are we going to be doing with this? Well, so now I'm going to be considering these space, these complex time surfaces, and they look like this. Now this is an idealized, this is literally an idealized model-based time surface where the radial coordinate from the center of the surface gives me time, and then the angular momentum gives me the corresponding time phase. Okay, so these are the objects that we are going to be looking at, and we are going to be asking ourselves, well, how can I actually estimate the time phases if I cannot observe them directly? So here is just another example, kind of illustrating how you go from a fMRI, functional magnetic resonance time series. Again, in these, you typically have for every voxel location in space. So I'm fixing the voxel for a second here. I have a fixed spatial locale, and then I have the fMRI signal dancing around, very, very high signal-to-noise ratio. And here is the top view and the slight side view of this fMRI signal. Now in fMRI, because the signal-to-noise ratio is so poor, typically 0.04, so the noise magnitude way outperforms the signal magnitude. So what we do there instead is we image under the same paradigm, under the same condition, many different times. So imagine the blue curve here and the red curve representing two different time courses of the fMRI signal for the same individual under exactly the same condition. So the experimental design is kept identical, and I have two different time courses. We aggregate these together, and now all of a sudden the entire repeated sampling for the same individual under the same condition becomes a Keim surface. Now these Keim surfaces, they look like this. So here is a location. So this is kind of looking cross-section right up here in this plane. I'm looking at axial or transverse cross-section of the brain. Here I'm looking at a sagittal cross-section of the brain. And right up here at the bottom right, I'm looking at a coronal cross-section of the brain. So when you obviously three planes intersect in a point. So here is this crosshairs give me the point. At this fixed local spatial coordinate, I actually have a time course. So this is the fMRI signal. What we are trying to do is we're trying to analyze it. So what we are going to do is there is two different paradigms here. The subject undergoes stimulus, rest, repeatedly multiple times. So here is the reconstruction of the on versus off Spacekime representation of these multiple time courses as a Kime surface. So they don't look necessarily very pretty. And again, on the website, if there is time at the end, I will show you, we can actually manipulate them in three-dimensional. This is just a snapshot of the three-dimensional image. And then this leads us to the point where, you know, we can learn on the previous 140 and then project prospectively, do prediction of the time course. And here the trace 7 is effectively the actual fMRI, and the forecasted time series is in red. The corresponding 80 and 90% confidence intervals are in gray. So that's what we are trying to do. Now, very briefly about Bayesian representation. You see, we can actually do this fairly effectively. Suppose you have a single space-time observation, xi0, fixed. Now, this specific observation comes from some kind of a distribution where one parameter distribution, and the parameter has the parameter has its own distribution, process parameter vector. So what we are interested in is we're interested in the sampling distribution P of x given gamma with respect to the corresponding phase aggregation and the corresponding observed data. So effectively what we are going to be doing is we are going to be trying to advance. Okay, there we go. What we are going to be doing is we are going to be formulating, using the usual Bayesian formula, the posterior Bayesian probability for the parameter given the data and some kind of a phase aggregator, some kind of a phase estimator or a model. We want to compute the posterior distribution of the parameter gamma. And the way this is going to be done is by essentially observing the evidence, right? So we're going to make this proportional to the likelihood and the prior. And once we have that, what we want to do is we want to compute two complementary probability distributions. The first one is going to be called the prior predictive distribution of a new test point, xj0. So I want to find what's the probability of me sampling a brand new space-time observation, and that probability is going to be defined using the prior distribution and by weight averaging effectively the sampling distribution and the corresponding prior distribution. The second instance is the posterior predictive distribution, of which I'm going to show you an example, which kind of gives you the likelihood that a data point marginalized over the posterior. What is that probability? And it's computed as the sampling distribution weight average by the posterior, not the prior as we did before. So here is just one example, perhaps, of this. So we are going to do a little simulation. We're going to have two sequences. Now they're going to be mixtures, as you can see, because U and V are mixtures of normal distribution with different parameters. So I'm going to have two sequences. I'm going to show you an example of these. And then what I'm going to do is I'm going to construct two additional cohorts by splitting the first cohort A into training data C and testing data D, because remember, I want to do some artificial intelligence here. So here are the 4 steps that we're going to be doing. Transform all these 4 cohorts in the Fourier K-space, in the Fourier domain, iteratively and randomly sample single observations from the training cohort, reconstruct the data back in space-time. So we're going back and forth as the diagram that I showed a little bit earlier, to see these reconstructions based on derivations, phase derivations from B, C, or D. Remember, C and D are different from B. B is a completely different process. It has nothing to do with A. C is just the training data for A, and D is the testing data, which is independently reserved for validation at the end. So right up here is the diagram that kind of shows you the two distributions that are very, very distinct indeed. And here are the corresponding reconstructions, right? Using the incorrect, totally wrong different process phases, you get totally wrong sample statistics. On the rows here represent different sample statistics, mean, quartiles, etc. So these guys are very different from where you need to be. But using the true phases, you're pretty accurate and pretty close to where you need to be. If you use D, the independent phases, you're also pretty accurately capturing the process. Now, in this specific Bayesian simulation, we can also see that we have a pretty good correlation by reconstructing the signal. So here's the correlation on the axis. I have the original data. On the vertical axis, I have the reconstructed data, and this is A versus C. Okay. And in this specific simulation here, we have an example where we're actually looking at 4 different sample statistics. So by sampling the posterior predictive distribution that I just showed you a second ago, here is the corresponding original distribution in dark blue, and your samples, some of them are bimodal, some of them are unimodal, you know, whatever it is. On the top right here, we have a bivariate test statistic. On the horizontal axis, I have the mean. On the vertical axis, I have the standard deviation. So you can see that my actual data, the dark blue, is kind of in the middle of the distribution. Another test statistic on the bottom left corner here is the maximum test statistic. Again, the posterior predictive distribution captures pretty well the actual value of my sample. And the last test statistic is the interquartile range here, the IQR, which kind of again demonstrates that you capture pretty accurately the posterior predictive probability distribution. Now, we can also do tensor-based modeling. Again, this is an example that you can actually walk through. Literally, the code is there on the website. It kind of shows you everything. So because our data now becomes intrinsically complex index, complex time, complex value, you guys, we need to use tensor-based analogs to the classical linear modeling. So we're actually fitting the tensor linear models, and we're estimating, so you can see the progression of the analytics, where here are for this finger tapping task, where the user is doing a finger tapping task. We can see exactly the temporal area, the somatosensory cortex in the brain light up when a person is doing this specific task, which indicates that we can capture pretty accurately, you know, the statistical modeling for this specific data. And in the last minute or so, I know we're kind of running out of time. Again, here is a link to the tutorials. TCIU stands for Time Complexity and Inferential Uncertainty. .predictive.space. You can look at spacekime.org. The R package is publicly available at CRAN and also on GitHub. And this is our team, as our esteemed colleague Gonzalo Abacassis always says, the secret sauce is teamwork. A number of my colleagues, you know, Milan Valef, obviously my co-author, students, Yu Yang, Dashuan, Zhijing, Yongkai, Zhe, and many others have contributed significantly to this work. So thanks very much. I'll be happy to answer any questions if there are any. And then the slides are available on the website. Mariana?
Sorry, I'm having a hard time finding the unmute. So please, if you have any question, you may either ask it on the chat, or you can actually unmute yourself and ask the question.
So maybe I can chime in and ask a question, Ivo. Ivo, very cool, very deep stuff. This is very nice work. I wanted so when you were towards the end, when you were discussing your the posterior. The posterior probability distribution. So you mentioned that you have the evidence here. So you have the probability of the evidence also. Usually in a lot of Bayesian uncertainty approaches, that is hard to obtain. But here you do have the evidence also, the probability.
No, we are, Krishna. This is a great question, Krishna. So what we are doing here is we're effectively computing the posterior distribution, and that's what. So these diagrams effectively show the posterior distributions because we sample from it, I believe, 2,000 times. We sample all these statistics. We sample these thousands of times, and this is what the light blue line is. The vertical dark blue is simply showing that specific statistic for a specific instance that we have observed. Okay. So, I mean, you don't want the distribution to be here and now all of a sudden the blue line over here, way off or something like this.
Yeah. No, I understand. So, okay, so the evidence is, I mean, here too, the evidence would be, the probability of the evidence would be hard, would be expensive to get, right? I mean,
Yeah.
Okay. Okay. So it's basically okay. So then the approach of scaling everything by the unknown evidence is the approach here, right? The unknown probability of the evidence.
Right. Right.
Okay. Okay. Great. Thanks.
Other questions from the audience?
So maybe I can chime in with another thing. So with the tensor descriptions, is there you mentioned it towards the very end, the last slide or the last couple of slides. So is there significant compression that you're seeking to gain here, or are there?
Yeah. So it's not really the compression that we're going over. So in this specific case, this was, I'm sorry, in one slide, there's so much.
I know, yeah.
So what we're basically trying to do, Krishna, here is we're trying, because you see, the data is intrinsically tensor-based. The moment you complex, because you see, you look in time, why everybody loves time? Because you see, you have a nice order. Time is longitudinally ordered, right? Everything is done. In time, there is no order. I know I kind of mentioned it in passing in the beginning. There is no order. And because you don't have order, your data samples are actually tensors. Because I cannot order them linearly. They don't go from 1 through n, right? They're kind of, they're kind of planary, and they may come in any specific order in the plane, right? So there is no natural order. So we have to do the linear modeling. The linear modeling we have to do on tensors.
Okay.
Right. And this naturally kind of leads to complexifying a little bit. And I tried to explain. I'm sorry, I hit this under the rug a little bit, but there is a lot of that goes into. First of all, the brain gets parcellated into 56 regions. The first tier of the analysis in finding out which of these regions are activated. In the second tier, we do localized analysis, but only in the active regions. And the third tier is we do post hoc correction using FDR and clustering. So we want to identify voxels that are not single, kind of single sparks. We're looking for a cluster of voxels that indicate some kind of a strong regional co-activation that corresponds with the person performing that specific finger tapping task.
Okay.
So the tensor is effectively a generalization of the classical, and it can be regularized as well, but in this case, it's just a simple linear model generalized to the tensor space.
Okay. Okay.
But the information you get from the fMRI is the same, right? It's just the way you are analyzing is different.
Yeah. Except that, Mariana, remember, we can actually go ahead and do these. Okay, so let me check. We can actually imagine transforming these fMRI time series that you see here on the left, right on the left. Imagine transforming them into this. So you're essentially doing manifold comparisons of manifolds.
Okay.
Right. Because these, you can use excursion sets, Gaussian random field theory, and all kinds of other things. So these time series, these time courses, which we do repeatedly over and over again, simply to be able to reduce, to increase the signal to noise ratio, because we don't have it, we're compressing them into a single Kime surface. And these Kime surfaces now, we can develop new techniques, and I haven't spoken about it, but you can develop new techniques to find out beyond which level is there statistical significance, and beyond what level this on versus off is just random noise.
Okay, and comparing time to analyze it, is it like computational time? How expensive
okay.
is it?
Yeah. So this is a great question, and I'll tell you, Yu Peng and some of my students, Rong Qian, they just cracked. We were having a problem. Okay, we have a significant problem because the Laplace transform that allows us to go back and forth between time series and Kime surfaces is difficult. Okay, because it uses path integrals and the numerical calculations were not very good. But we are right now releasing a new version of the package that's fairly expeditious. A couple of seconds to do one of these things. So there is an added computational complexity. You probably double or triple the calculations it's not a 10-fold or 20-fold or 100-fold. The calculation increased because of this added on necessity of Spacekime representation of the data before we even start anything else.
Right. And it also depends on the number of phases you use, and I guess many other variables.
Yeah, exactly.
Okay.
Yeah, exactly. The sampling, the phase space, that does matter, and we have strategies for this. Yeah.
Okay. But in theory, if you have your own data, you could use your.
Yeah.
Okay.
If you use your, if you know, right up here, if you know, I mean, you know, we have all our code is R based, and you can see, so right up here, if you click on the code, you're going to see all the code.
Okay.
Anywhere in this tutorial, you know, when you have, so here is the actual fMRI signal, right? So you can actually see these things in action, and here at the very bottom, you can see, right? So here is the difference Chime surface that I just showed you a second ago. So this is the, as it says here, fMRI on-off difference. Okay. And then the most important thing, of course, is some of these guys, and that's ultimately what you're looking for. You're ultimately looking for these guys. Let me just show you right up here. Right up here, you want to eyeball. You want to eyeball these, you know, these brain locations, and you want to say, aha, this is exactly the motor area bilaterally on both hand sides, right? Because through a cross projection, right? If you have one static image like this, you don't really know where it is. But once you have it interactive like this, you can actually pair it through and see how the original p-values, after the correction, they go into phase 3, they shrink, right? They shrink.
Okay. Oh, that's very cool.
Yeah, and all the code is embedded in here. Yeah.
I don't see more questions from the audience.
Hi.
Oh, okay.
Yeah. Hi. Thank you for the very wonderful talk, Professor Dinov. I was just wondering quickly if, so I was wondering how this would translate in terms to how data hungry the approach is. I mean, would it, if we have very small data sets, typically it's a problem to work with fMRI data, but would this approach provide any particular advantage in small data sets? Or
Yes.
would it
So,
be the other way?
Divya, this is a great question, and Krishna, I mean, I'll tell you, this is really controversial, but I'll tell you my interpretation. So Divya, the most important ramifications of this is, you see, that if you have a pretty decent estimate of the Chime phase distribution, and I haven't shown you any of these, but I think I might have some these Chime phase distributions. Yeah, I don't have them here. If you have one of these distributions, you see a magic thing happens, because you can actually go ahead and randomly generate data that's really, really realistic. Why? I tried to hint to this early on with my example of the situation of having a single observation and then trying to sample from the phase space. But Divya, you're absolutely right. fMRI, they're huge volumes, they're expensive, they're difficult, they require patience. You know, the point I'm trying to get across is that if you get 20 patients that are Alzheimer's, you image them, and then from these 20 patients, I can make 2,000 very, very easily by guesstimating somehow, and we have strategies for this. We have Laplace transform analytical strategies. We have phase aggregation strategies. There are estimates by which you can estimate the phase distribution. If you have the phase distribution, you can amplify these 20 individuals, Divya, into 2,000 individuals instantaneously. So, yeah.
Yeah, sorry. So you mean that once we get the Spacekime transformation, then we can sort of generate our own distribution of the population?
Yeah.
Okay.
In fact, we go from IID random sampling in the observable phenomena. We go to Spacekime sampling, which is computational completely. But again, there is a big if, because you still need to know or have a prior or an oracle or some kind of an aggregator to be able to say what should those phase distributions be. This is a big question. It's a tough question.
And to obtain those phase distributions, do we need a lot of data?
Well, okay. I'm sorry, I don't have an answer to this question simply because it's so subjective. If you want, I can dig out. We have studied many processes, and the phase distribution space is, as you can imagine, quite complex, right? I mean, for one process, right up here, I'm not sure if that will be visible. For one process, it may be simple, just like here, right? You see, for some process, it may be this distribution. For some process, it may be that distribution. For some process, maybe that. So the variety of distributions, are going to be subjective. So I don't have a means of dictating for some experiment what will be the right distribution. So, for example, Alzheimer's disease individuals might have this phase distribution. Mildly cognitively impaired individuals might have that distribution, and the normal controls might have that distribution. But again, experimentally, we can try to guesstimate them.
I see. That's very interesting. Thank you.
Yeah.
Any more questions from the audience? I guess it's 12:02 already. So thank you again, Ivo, for the talk. It's very interesting and so much information.
Very impressive work, Ivo.
Yes.
Thanks very much. I appreciate the opportunity. It was fun.
Yeah.
Talk soon.
And thank you everyone for attending. Bye.